So now we are going to discuss macromolecules. We've talked about functional groups, we'll bring them up again, and now we're going to build larger molecules that will basically carry on all the functions of the cell. So the macromolecules are built of pieces. Oftentimes they're repeating pieces called monomers, which we'll go into in detail. So sometimes it's a little bit of a modified monomer, and we'll get to that when we go to each section. But in any event, all the macromolecules are put together with the same type of chemical reaction. And basically this chemical reaction to put the pieces together, here they show you a few pieces already put together, a short polymer made of several monomers. We're going to bring in a new monomer here. The first one that we'll talk about will be carbohydrates, so let's call this glucose maybe because we're building a glucose polymer. So what happens is an enzyme needs to facilitate this reaction because we're going to rip a hydrogen off that growing chain and also rip an OH group or a hydroxyl group off this incoming monomer. It's called a dehydration reaction because basically you remove water, you dehydrate the water. So enzyme brings that in, we create a bond between the two monomers, and we move on. So the opposite of that takes place, for example, during digestion, and that would be hydrolysis. So when you eat maybe a potato, you bring in a lot of starch. You'll see some of those in a minute, those molecules in a minute. Basically, they're a huge polymer of glucose. Enzymes start working first in your mouth, amylase, and then later on in your stomach, anyway, other enzymes that chew up the carbohydrates, and they're going to come in, bring a water molecule in, so it's called hydrolysis, and split that water molecule and add a hydrogen to the chain and then an OH or hydroxyl group to the monomer, break it apart. Now you can go use this piece. So what are these macromolecules? Well, we're going to focus on 4 classes of macromolecules. Today I'll talk about carbohydrates and lipids. You'll do a few interesting experiments with these guys. Broken down in this slide is what the different macromolecules are built of. So again, we're going to focus on carbohydrates and lipids. There are many monomers that carbohydrates are built of, and we'll just focus on glucose and fructose primarily. Galactose is in there because it's an isomer of these 6-carbon. We'll talk about some other monomers as we go on too. There are dimers in the carbohydrate world, and you know that as table sugar or sucrose. We'll talk about that a lot as the year goes on. Polymers are basically thousands of glucoses hooked together in these cases. Starch and cellulose, we'll explain the difference between them, and we'll bring up the idea of glycogen briefly. The second macromolecule we'll talk about today are lipids. They break the rule of the monomer-polymer idea. They're basically a glycerol molecule that you'll see a picture of in a few minutes, and then fatty acids that get attached to that. Or in some cases, they're a glycerol with a phospholipid and fatty acids and a special phosphate head group. Again, we'll talk about all those as we move through the presentation. So first, let's talk about carbohydrates. You know carbohydrates as that super energy producing food. It's sugars, it's starches, it's potatoes, it's bread, it's candy bars with sucrose in it, or that dreaded Coca-Cola. You get a lot of energy from it because we break down the glucoses that some of these are made out of and feed that into energy generating pathways we'll talk about in a few weeks. There's bunches of different lengths of carbon chains. First, let's talk about some smaller ones. So a triose has 3 carbons. This one's brought up over here because it'll be an important intermediate during glycolysis and also during photosynthesis we'll talk about. Note up here that functional group. What is it? What is it? What is it? That's a carbonyl. It's at the end. So these are all aldehydes. Then a pentose that you will become familiar with is ribose. If it's missing an oxygen in I believe it's the 5th position, it's maybe the 3rd. We'll see a picture of it later. It's deoxyribose, and that's a component of nucleic acids. Ribose is an RNA. Deoxyribose, we'll see it in the next screencast, is a component of DNA. Then we can go to the 6-carbon sugars. Hexoses for 6. Famous glucose, C6H12O6, and then fructose. Now these two are isomers. What kind of isomers do you think they are? They have the same molecular formula, but they have different structures. So they are structural isomers. And this is the difference of of different carbonyls. Remember, if it's at the end, it's an aldehyde. If it's in the middle, it's a ketone. And also, they got tons of oxygens and hydrogens or alcohols hanging off it, so they are definitely alcohols. We're going to spend lots of time with glucose over the next couple of weeks. So, glucose gets that magic size length that it has 6 carbons, and it is most thermodynamically stable to condense into a ring. You don't gain or lose anything, you simply shuffle things around. So again, remember, in organic chemistry we label our carbons starting at the top 1 through 6, and then here they are again, 1 through 6, and they keep the color coding here, which is a really nice help. So it is thermodynamically stable for this double bond to oxygen to bounce off and steal that hydrogen. That leaves this oxygen needing a bond, and it'll go make it with the carbon who now needs a bond. So again, move that oxygen bond out and grab a hydrogen. Now this oxygen will attack that carbon, and the OH becomes down here. So this is a very stable form of glucose, and we're going to see these little green hexagons all over the place. In fact, most of the time we're going to drop the nomenclature that has the C's at the vertices and the O there, and just know that when we've got this thing here, it's glucose. You'll see it coming up soon. Here's a little bit more of an abbreviated ring structure with the carbons missing, and the oxygens and the OHs are here. Although if I draw it up, I probably won't draw those up for you on the board. Whoops, did this already. There we go. So now, which molecules do we want to look at? So I did not go through how the fructose condenses in a ring, but just suffice it to say, because of where that ketone is now, that double bond to oxygen makes this a pentamer, or a 5-membered ring. It didn't get rid of any carbons, it just shoved one out here and one out here, whereas glucose has an OH down there and a CH2OH up here. In any event, it's the way we can tell the difference between glucose and fructose. If you want to do a condensation reaction, remove a hydrogen from fructose and OH from the glucose, make a water molecule. We then make a bond between this carbon here, who is needing a bond, and that oxygen. You can see that down here. That's called a glycosidic linkage. You don't have to remember that so much. And we make a bond. This is now called sucrose. This is what table sugar is made of. So table sugar is made up of a glucose and a fructose. Oops, did that. There's sucrose. Sucrose is not so good for you because fructose is not so good for you. We'll get into that another day. So remember, those repeating little green hexagons are our glucose molecules. So what they're trying to show you in this slide here is that there are polymers of glucose called starch that occur naturally in potatoes. And basically, when we eat a potato, we are getting thousands upon thousands of glucose molecules hooked together. We have enzymes to chop them off one at a time and feed them into an energy generating system called glycolysis. Animals store glucose to be ready to be used in emergencies or just basically for energy, and animals store them as glycogen, not starch. We make a different, we just coil it up differently, we make it a little more branched, and we store them a lot of times in muscle tissue. So that's glycogen. It's just a slightly different arrangement of glucoses, but it's glucoses arranged nonetheless. Polymers of glucose that are found also in plants, but we don't digest them, and I'll explain that in a second, are called cellulose. These guys are very stiff. Notice it's not all coily like the one we have in the starch. They're very stiff chains. These get put together basically in these fibers, and they make up the cell wall, microfibrils in the cell wall, and that's what makes plants stiff. There's a few things else that make plants stiff, but this is one of them. I'm going to show you the difference between starch and cellulose and why we really don't digest grass. So remember we said that the glucose is most thermodynamically stable as a ring? Well, when it goes to make that ring, this OH that's now going to be hanging off the first carbon has a 50-50 chance of either pointing down or pointing up. And that's just simply 50-50. So if it's pointing down, it's called alpha. If it's pointing up, it's called beta. But what's the difference? Well, if you put a linkage, you link glucoses together of alpha glucose, you make starch. And we can digest this bond because our enzymes recognize the shape, and we can cleave it here and we can use our glucose. If glucose is put together in beta monomers, glucose monomers are hooked together. So notice there's an OH pointing down, now there's an OH pointing up, and it's because of the way it has to flip when it makes the bonds. Don't worry about it more than that. We don't digest this. Our enzymes cannot recognize this shape with the oxygen, the hydroxyl pointing down and that hydroxyl pointing up. And we don't digest this. This is why, like, we don't eat grass. Or, to get a little graphic, when you eat a corn kernel, the kernels come out. So there's an up close and personal picture of those two glucose structures. And who can eat the grass? Well, the cows can. And it's actually not the cows who are eating the grass. It turns out that the ruminants, cows are members of them, have multiple stomach chambers, and they have bacteria in their stomach. We have bacteria in our stomach too, we just don't have the same ones. That bacteria is specialized to cleave this 1,4 linkage of beta glucose molecules. And they can lop off those glucoses, and they can get energy out of eating the grass and all the other straw and stuff like that. We can't do that. We have the cows do it for us, and then we eat the cows.